Hello, everyone. This is the next episode of For All, the webcast of our Biotech Alliance. My name is Leif Kruse. I'm CEO of For All, and with me is Puya, CEO of Risklick. Risklick is a tool, if I understand this correctly, it's an AI tool that you use to optimize clinical trial protocols. AI is sometimes quite a buzzword, but I think you have some kind of real application that you're using to support biotech companies. So welcome, Puya, and maybe you give us a little bit insight of who you are and what your company is doing and what this AI in general is about.
Hello, Lloyd. Thanks for the kind invitation and happy to be here and discuss this really interesting topic with you, which is really close to our heart. And this is what we are doing as core of our business. As you mentioned, Risklick developed the first data-driven solution to design and optimize the clinical trial protocol. We have an approach and the data-driven solution to empower the expert. We don't want to replace the expert because what we believe is that the solution and tool, these are the tool to empower the expert, and only the AI work in the combination with the expert. And that's the perfect match that can really increase the efficiency of the protocol design and optimization process, because the expert, they have vast experience from the past, and they have a lot of experience which is valuable. And then if we can empower the expert with simplifying for them to use or the historical data to give them a guide or give them some insight, then would be fantastic and works the best together.
Okay, thank you very much. I mean, this already answers more or less one of those questions that all people have: is AI replacing us? First, and second, how is this really going to work? Is really AI doing all the work? But I understand that it's more a supportive tool for experts to be more efficient in that. And I think we go into the process later on, how this really works and how you're supporting clients with this application. But first of all, let's discuss about why are we going to optimize clinical trial protocols? I mean, isn't this what we are getting from the experts already optimal? And if not, why is that so?
Yeah, that's a really good question. Going one step back, I mean, we need to understand how important is the protocol for a clinical trial. As you know, the most important document for a trial is the protocol, which we need to execute everything based on that. And we need to I think what we believe that it's a really sensitive document, because it is like a really sensitive software or IT system that we all know that is garbage in, garbage out. And this is how we see also the protocol for the clinical trial. If you don't have a good protocol, you can't expect to have a good result, because the result are the reflection of what you defined in your protocol. Therefore, whatever that's just want to share with you our view that how sensitive is the protocol. Therefore, we need to be really also careful and sensitive for each component we add or delete or change in the protocol, because we need to understand that has a direct impact in our result and outcome. That's one point. The other point, I mean, if you look at only the statistic and the publication regarding the quality of our clinical trial protocol in general, there are several publications shows that on average we have 3.5 amendments in the protocols. And as you know, each amendment delayed the project on average for 3 months and cost. Depends on the size of the project. And there's a really interesting study that showed 40% of the amendment happened before the first patient enrolled, which clearly shows that how is the quality of the protocol and so on. And from our experience, we see that usually this protocol development is really unsystematic approach and heavily rely on the expertise of the people. And since we tend to like or dislike some project based on our opinion, we have a selection bias that we take one protocol, try to replicate or use the information from a specific one and so on. That's why we think that there is a lots of room for improvement, for the improving the quality of the protocol, which is understandable because it's not easy to write a protocol. It's a complex and lots of people with a different expertise, different language are involved to develop that. And this is really difficult. And how much we can improve, I mean, there are just some publication that's an estimation, but it's not a representative of the whole study in the world. But they took some trials, look at it, and they estimate 50% of the failure is independent of the compound or medical device and can be avoided with a better design. I wouldn't say that all can be avoided with a better protocol design, but the protocol design is a big part of this avoidable amendment and mistake by going to a direction of the critical thinking. This is what we want to implement.
Okay.
This is that we want that the tool, AI tool, to help us to go to the direction of the critical thinking.
Okay, so let's quickly discuss, how clinical trial protocols are developed right now and what you're going to change in this process. So how are protocols developed right now? I mean, you already mentioned it's more or less something on the fly. So we have a nucleus which comes from, I don't know, historical protocols that we know from competitors, clinicaltrials.gov, maybe other trials that we have conducted in our company. Are there any other sources where clinical trial protocol development starts?
At least to our experience and knowledge, and then the knowledge of our customers, some of the big pharma that we are working with, the way it is that we first start with selecting one protocol of the similar study from our friend, organization, or the one that we like the study, and start to copy pasting some part of it to make the core of to make the concept sheet or synopsis of our protocol. And then from there we involve the expert to give their opinion. We go to the key opinion leaders to get their opinion and integrate and all these opinions and feedback and develop a protocol and optimize the protocol. Of course, then there in a lot of company, there is a quality management system in place that somebody at the end check that all the protocol, that all the sections are, are in line with each other. But to our knowledge, that's the way that we are doing and lots of other people are doing.
Okay, but where's the problem then? I mean, I'm working with best practices, with protocols that worked, and showed very great success in previous trials. Makes sense to base on that or not?
I mean, it's really important to consider that, but looking at only at one protocol or two protocol as a basis for development of a protocol is a little bit dangerous, because our setting is really specific. Is not the same. We don't want to reproduce exactly what they did. Yeah. I mean, we want to innovate, and the trial is always about innovation, because we make a new project, new idea, new intervention, new indication, and so on. And then of course is not the same. And then if we want to copy paste only from one that is similar but not completely the same, but repeat what they did for with a new setting, that would be a little bit dangerous.
Okay, I
That's
got
not.
that. So if I understand you correctly, with your AI tool, what you did is you have, trained this artificial intelligence in looking at all available protocols. Would be interested in what sources you're using, if you can reveal this. And but if I get you right, with your tool, we have a broader view on many more potential protocols to base on. Is that what you're saying?
Yeah, exactly. This is what we believe. We believe that we need to look at systematically all, not select one or two, which have, first of all, we have a selection bias, and this one or two is not a good representative of the thousand. As an example, in some cases we have 10,000, 20,000 and more, but it's not a good representative. And when the advantage, when we look at systematically the whole studies, we can find the trend, which is not possible to find it by looking at 1-2 study.
Got it.
That's how. And then the other advantage is that when we look at systematically the whole data, we can decipher, I mean, what is good in general that most of the study have done and they were successful, and what most of the study were failed with. That's
Okay, let's talk
that.
about input output.
Yeah.
And I understand your input in your with which you have trained your artificial intelligence is all available protocols that are out there in the universe. And so what are we then now putting into this AI, and what is the output of your tool, and what are you doing with that? What are you optimizing then?
Basically, we empower the expert to ensure the quality of the clinical trial protocol from the time they have an idea until the end of the study. That's our aim and purpose. How we do it, we also believe that we need to develop and optimize the protocol step by step, because it's a complex system. If we want to handle all the complexity at once, that's too much. Therefore, we need to break it down to smaller components, look at each component to make sure that this component is good for our setting, for our trial, and then put all these smaller components together to build a complex system that we call it clinical trial protocol, and then optimize the whole complex system to make sure that all these good selected components also working well together, they are in a streamline, and so on. Therefore, the input can start from the time that you have only the idea of the intervention and condition to give you an overview of the space. I mean, okay, how many trial running, what is the main probability of success, what are the main obstacles that they are all facing, and so on. Really broad overview of the space, and some general information how the space look like in the trial. And then you get the idea, okay, I want to do this type of the trial, and then give you a recommendation. What are the most common endpoint in this area that the past study used, were successful, failed, and so on? What is the most common patient population in terms of the eligibility criteria, the other use, and so on, to give you some ideas where to start systematically. And then you take that into the account and start building that step by step based on the data. And then when you put all these pieces together and create the first draft of the protocol, then you can enter the first draft of the protocol, the concept sheet, synopsis, or the first draft of the protocol into the model. And then the model consider all these points, that's how they are working together, if it's a good combination, bad combination, or they are risky or not risky, and how to mitigate them.
Okay, I got that. One thing that I still don't get is, and maybe you can give me a little bit more insight in that, how are you evaluating whether certain parameters, are good or bad in terms of selecting them, but also in terms of what their value is? How do you evaluate this with the system? Does the system evaluate this? And if so, how? And what is the output?
Yeah, it's really good question. I mean, that goes first, that how we define the success and failure. That's a
Okay.
big question. And then we already define the success and failure of a study based on the several factors, not only based on the factor that they are completed or terminated, based on that if they could met the primary outcome, if they could enroll the patient on time, if they could finish the study on the estimated time, and dropout rate, and many other factors. First, to label all this historical study to if they were successful or not. That's the first step we have done, and we regularly do it with the new studies as well. But when we look at your study, for example, and then the model look at that this, for example, single inclusion criteria from two aspects, from the aspect of the intervention and from the aspect of the condition, that how many times this inclusion criteria appeared in the historical data from the condition and intervention perspective, and what was the success rate of the study who use such a inclusion criteria from these two aspects. And this is, as I mentioned at the beginning, this is just to shade a light and highlight the part and give some idea to the expert, okay, this is good or bad, and then the interpretation of this data, okay, then is it really good, bad, or this is in the hand of the expert.
Okay. So if I understand you correctly, you evaluated the different parts of a protocol, inclusion, exclusion criteria, geographies as well.
Yeah.
Yeah. Okay. Indication, and then give us an idea of what are good parameters and bad parameters in terms of which of those appeared in protocols with higher or lower success, meaning giving us an idea of what we should consider as good or bad parameters in the future, right?
Yeah. And the output is not really absolute that we say good or bad. It's really relative based on the data, because everything is a data driven. Sometimes it appears as in a bad category because it's so novel, but it's not bad. I mean, it's not by definition that nobody used that doesn't mean that's bad. Means that it's novel, but then that's a consideration to inform you that nobody used that. Are you aware of that? Okay, yeah, I know, but I want to try that. Doesn't mean that you fail.
Okay, I got that. So we have now a kind of combination that the system advises in using. Now we are building up the first draft of our protocol, and we then put it back in the system again, and the system does what?
The system specifically, we look at the most important part of the protocol that is very important for the outcome of the study, which one of them is the endpoint and outcome. As an example, the system check, okay, how standard is this primary outcome that you are using? Everybody use the same primary outcome? Mm-hmm. Or the primary outcome that you define in your protocol is new? That's also a flag that say, okay, how the regulatory bodies will accept that new primary endpoint that I want to measure. Or because as an example, we had a case last week that they want to use a really novel endpoint, and then of course in the analysis flagged risky because nobody used such an endpoint. And then when they discuss it with the FDA said, yeah, true, this is not clinically relevant for the patient. And then even if you complete your study, we cannot approve your medical device for that claim and that indication. Mm-hmm. And these are really important issues that we need to critically think about them and then optimize them before we start. And then the eligibility criteria, inclusion criteria, exclusion criteria is another part. We also look at the processes regarding the patient burden, because what we usually tend to do as a scientist, and I was doing the same when I was writing protocol, I was sitting in my office with my colleague and imagining, okay, what I want to measure. I want to measure this. I want to get this data. Yeah, I want to analyze that. I want to understand, because we are curious to know what is the correlation or relation of this drug to that adverse, and so on. And we want to measure everything. But we need to be careful that then the patient and the volunteer would like to or should participate in this study, and how heavy, how difficult is for them to participate in a study. Therefore, we look at the patient burden. We look at from the perspective of the patient, how difficult is for patient. And as I give you a concrete example, we had a case that they want to take the blood from the patient 20 consecutive days. And considering these people have a job, they are working, family, they are not all living close to the center, and they had to come to the center to take the blood and so on, then that was a flag from the analysis, and we challenged the team. Do you think that somebody come 20 days to give a blood because of the study? Mm-hmm. Do you give that much advantage to the patient to motivate them that spend 2-3 hours per day 20 days to come and give a blood? I know.
Makes sense to me. Yeah. So that this is, where I understand that your tool is evaluating the protocol scientifically for scientific success on the one hand, but I think also for operational success. Do we have a protocol that is really operational, executable in a way of, are we capable of recruiting enough patients? Will this be feasible for investigators and site to run this protocol? But also, don't we have too much dropout rates because patients say, yeah, nice drug, nice therapy, but nothing for me because it doesn't fit into my daily life schedule. Okay, very interesting. Yeah. So that means, and that brings me to two other questions, or one question with two aspects. What is then that the system produces? Is it kind of a report? Is it? I understand there is some kind of heat map where we can see, okay, this is more in the green, in the green light. There's something in the red light and something in between. And what do you think also about having this tool being, I would say, a little bit neutral when we have, those sometimes political discussions with key opinion leaders where they really have, those exploratory endpoints or measurements in the back of their mind, which is nice, but maybe not must be.
Yeah. The output is including lots of heat map and graphs, which at the end come to a comprehensive report, which is really easy to read, because if you look at, for example, in a case of the eligibility criteria, that would be that is a heat map, that when you look at the heat map, you will see, okay, my inclusion criteria number 1 is green, fine, but okay, number 9 is red. Okay, now I need to go to the detail of number 9 to understand why is risky. And this is one of the advantage of our solution, that we call it explainable AI, because it's important to know why it's risky. If you show me that it is risky, I mean, it's interesting, but at the end, if I want to resolve that, first I need to know why, and based on why, then I can define my mitigation action. And this is how we do it. We can show the expert why the model calculated risky. Okay. And then how they give them some idea how they can fix it. But this is, as I said, this is a recommendation. We leave that to the expert. We want to stay neutral, unbiased. They say, this is what the data is saying. Then the interpretation, or how do you want to define your mitigation action, that's your expertise and it's yours.
Which brings us to the saying that I see in the back of you on the wall. What is the saying?
Yeah, it's saying that without data, you are just another person with an opinion. And this is not a phrase from me. This is a phrase from Edward Deming, which is a really famous statistician, which believe in a data and say that's the data should speak.
Okay, so the data and your AI is supporting the experts, but in the end, it's the expert to interpret the data or the outcome of the data, the outcome of the system, and make some informed decisions about how to optimize the protocol. So you're not optimizing the protocol, you're more giving us an idea of what might be risky and what not, and what the probability of success could be if we do this or if we change the protocol in the way that the system advises, right?
Yeah, exactly as it is. And coming back to your previous question, because you nicely mentioned the discussion with the key opinion leaders, and this is how we believe. We believe that you need to have a good ground of the data, and then you can take it and go to and discuss it with the key opinion leader and enrich that from there, because now you have a ground that as a not field experts, then we empower you that you can negotiate and then talk scientifically and also challenge the key opinion leaders. I mean, they are expert of the field, but then we know that the science is all about the discussion. Mm-hmm. And mm-hmm. Then okay. This is, we want to bring the people that not just to accept naively the opinion of the expert because they are expert. We want to challenge them. We want to discuss with them. Okay, you propose that. Why? Because the data is showing 48 hours we want to measure, but you want to go for 24 hours. Do you have that scientifically a backup for that, or is just a subjective opinion?
Super interesting also for us when we are looking into the costs and the overall burden for biotech companies when they are executing their trials. Definitely optimizing clinical protocols is the way to go if you want to reduce costs and conduct clinical trials more lean, more quicker, and with less costs. Can you give us some ideas of business cases that you have in the past. Have you done this? I mean, there's a big promise behind that, what you're saying, but can you also show this and prove this in best practices, in business cases? What was the outcome?
Yeah, that's we had a very interesting case because that was an investigator-initiated trial that the sponsor had a contract with a pharma company to conduct the trial for, and they had a budget, they had an offer from a CRO, but somehow before they start the study, there was an investigation on a CRO for whatever reason, and the sponsor decided to cancel the contract with the CRO. And then the sponsor went to the other CROs and asked for the offer. And then, of course, all the CRO gave the bigger, the higher offer that was not in line with the budget. And the guy was said, okay, nobody execute my study with this budget. What should I do? And then contacted us and say, guys, can you help me to see where can I cut from my study to make it feasible within the budget I have? That's all I have. I can't increase it at all, but without losing the science part of it, without jeopardizing the main outcome of the study that we are interested in.
Mm-hmm.
And we did that with them. And then what we could do, we identify some of the secondary outcome which were not really necessary for the main purpose of the study and which had a big influence on the cost.
Yeah.
And then we identified that part, and then we informed the sponsor, guys, these are the place we think if you cut it, you don't lose the main purpose of the trial, but you can cut the budget and optimize it. And it was, at the end, it was really good because they could make the study also lighter for the patient. And then the guy said, yeah, we want to measure. It was good to have, but we didn't want to even analyze this data. We wanted to collect this data to see what we're gonna do with this data later on. Mm-hmm. Which is really interesting. I don't remember that, but there is a publication that they showed how many percentage of the data in a trial that we collect, but we never analyze.
Yeah, classical situation. Nice to have,
yeah.
Nice to have in the protocol, and driving all the costs. That's what we see as well. When we are analyzing protocols and see where we can optimize the trial from the operational perspective, cost burden. Love that. Okay, so in the end, this trial has been executed in budget.
Yeah.
Cool.
Now they got the agreement with the CRO that changed. They communicate to the CRO that they changed the protocol a little bit, and that they believe that now it is within the budget, and the CRO confirmed that. They signed the contract.
Budget is one thing. Success probability is the other thing. Have you also some kind of business case around that, maybe retrospectively, to see what if we would have done that differently?
Yeah, this is really interesting question. We have done several retrospective study to challenge us, but to see, okay, but we are claiming this is real, or this is no, is a nice to have. Mm-hmm. And is a luxury, another luxury tool that is nice to have, but is not really necessary and doesn't have any impact. What we did a retrospective study in a way that the sponsor gave us the protocol of a study that started 3 years ago, and they gave us the first version of the protocol. And of course, they knew all the issues that happened during these 3 years. And they asked us, based on the first version of the protocol, to predict the risk and say where we suggest to optimize or change and what would be the recommendation. And then to compare our prediction or with the real issues that happen to see how many percentage of that match and then how many percentage of that we could mitigate before the start and what does it mean in terms of the money and cost. As a result of this study, we could show that we could predict 75% of the issues correctly. When we quantify that in the money and time, it correlates to 2 years and 40% of the budget.
Which,
Means if we have done this approach and this analysis before they start, they could save 40% of the budget in 2 years' time.
Interesting. I mean, yeah, it's good to have this, data where you're challenging yourself to see if the model, if those predictions really turn to be true in the end, because it's always good to be wise at the beginning, but see if those changes really apply in efficiencies in the future is good to know. Interesting.
Yeah, because what we believe, we are trying to challenge our clients to push them to the direction of the critical thinking, and the same we have to do it first on ourselves.
How's this now going to work? I mean, is this a SaaS software that I can just use in the internet? I put my protocol in there, and and I get the output, or is it that we are collaborating, that clients are collaborating with you in optimizing this? How does the process look like?
Yeah, the solution is a web-based solution, is a software as a service solution. But going back to our earlier conversation, AI is just a solution, is not a magic bullet that we can put and get the protocol out. It's an interactive process by using the solution to optimize a protocol. And the way that we do it, we give access to our clients, and then is a web-based software that they receive their user password, they can log in, and they can choose the 2 options, the assisted option or self-service option. The assisted option, we help them to make sure that the data entry is correct, or if they have any questions scientifically from the clinical trial or medical aspect or IT aspect or data science aspect, we support them for the data entry. We do the data entry together with the customer. Once we are both happy with the data entry, then we run the analysis, we get the result, and then based on that result, we might go back to the entry, change something to see how it changed, and then take it from there and interactively, step by step, with the expert and client to do it. And it's a process. It's not one-time analysis that you shoot it and you get the result. That's how it's. But then our aim is that we are working with our customer to also train them on a system and enable them that they use this solution without us in case they have this expertise in the team. For example, in the case of the big pharma, they have all this expertise in-house, and then we are trying to train them, enable them to use it. But in case of the medium-sized or small biotech companies which don't have the capacity in-house, we support them from Risklick team.
Very good, very good, and I totally understand where you're coming from, and that this whole process is absolutely beneficial from the operational, from the scientific, but also from the cost perspective, where we're working together to get, budgets that we can then, and trial protocols that we can then complete in time and in budget, because there are less surprises that we face in clinical trials in the future. Very interesting. Thank you very much for that. So I think we have pretty much covered everything. I see the value in that. I'm really curious to see, how your company is evolving over the next couple of years. I think you're currently a team of, how many people are you?
We are a team of 15 people.
15 people.
In the company. We have a really multidisciplinary team because it's a really multidisciplinary project. We have doctors, pharmacists, clinical trial expert, data science, software developer, and so on, which are all working together to make it happen.
And I think you're also capable of supporting clients in all the regions. So you're based in Switzerland, but I think you can also support clients in the US or anywhere else in the world. So this is this is the advantage of IT today, right?
Yeah, exactly. We have some clients in the US that we are working with them because anyway, even if we are here in Switzerland, both and close together, we are working on a solution which is the web-based, and then we need to work on the solution together step by step. That's why.
Thanks, Pooya. Thanks, Pooya.
Just...
I really enjoy our collaboration in this matter, and I think that's what you're providing pretty much is a nice puzzle piece that we can also use in supporting our clients. So thank you very much for introducing us to some kind of AI tool that we really can use in protocol design, which is really important for our industry.
Thank you very much for your invitation and this nice conversation. I really enjoyed the conversation and happy to support you and the client in this journey, because the patients are waiting for new treatments. Whatever we can do to bring the treatments faster to the patient that they need, that's our passion, because either us or one of our family would be the patient some point, or are the patient now waiting for this treatment that are developing in a clinical trial. If we can make it faster happen, that they can have access to these new treatments, that would be to benefit of all of us, I think.
Okay, nice closing for that. Thank you very much, and see you then in one of our next episodes.
Thank you very much, and have a nice day.